All right. Well, hello, everyone. Thank you so very much for having me here. I'm very much a freshwater ecologist. My training's in stream ecology, and I appreciate the opportunity to be here. It's been a really interesting conference for me in getting the perspectives that you guys have, and I kind of have the same sentiment as Bob when I say, you guys have a different way of approaching questions, and so hopefully the things I have to say today are also interesting to you, and you can think about how you can apply the stoichiometry framework to the work that you're doing. And I just want to acknowledge several collaborators and opportunities that I've had in which I've been able to explore stoichiometry and think about how it influences the systems I work in. And got Bob up there at the Wood Stoic Conference, so if you ever have if you're an early career person and you want to talk about stoichiometry and also tie-dye t-shirts and have a dance party, I suggest you look into Wood Stoic. So as an ecologist, I'm really interested in thinking about how does biodiversity and abiotic processes interact to influence ecosystem function. Because we're all interested in what's going on across the systems that we work in. We're interested in the primary production that's occurring, how much decomposition, what's the carbon sequestration of that system. And I'm using this kind of framework to understand how are abiotic processes interacting with biodiversity. So what's that species richness, what's the composition, what's the age or size structure of that system, and how does that interact to influence ecosystem function, and how does ecosystem function feed back to influence these kind of things. And so we need to do research across various spatial and temporal scales. And from the work that I've seen here, it's obvious that you guys are doing a really great job at looking at large spatial scales to think about your questions. But we need to know about our systems. We need to have a strong understanding of the physiochemical structure as we're changing temperature, precipitation, and all these different metrics across the globe. We need to understand our systems and understand what's the tolerance of the organisms living in our systems. What are their traits? How are these systems going to interact with these changes? And how is that going to influence things like community structure and ecosystem function, things that we think are really important. And so I like to use this framework of ecological stoichiometry to try to understand these processes. And because we can go across scales to understand how is the organism being affected by changes in its abiotic template. So we can first think about, the resources. So, Tom covered this quite nicely. It's really hard to understand what organisms are actually ingesting. And as these resources are changing, it's going to influence what these organisms are provisioning back to the environment. So the excretion and the ingestion of elements is thought to be coupled to that ingestion. And animals are typically thought to be homeostatic in their requirements. And so based on what they require, that's going to influence what they're excreting and adjusting back to the environment. And these rates, of course, scale with temperature. And as we're changing the nutrient levels of these systems, these kind of rates are going to change. And this varies across taxa. And so we can think of the direct effects that consumers are having on nutrient provisioning via these direct effects that I just talked about and Bob covered really nicely. We can also think about the indirect effects that organisms can have on consumer-driven nutrient dynamics. So for example, Helena and Schmitz and this nice work done in a terrestrial system showed that in the presence of a spider, grasshoppers changed their grazing activity. And so they were feeding on food resources that were of a lower quality, of a higher C to N, and that also altered their tissue stoichiometry and what they were excreting as far as adjusting, that is. Furthermore, that this has been shown in aquatic systems as well. Costello and Michael showed that in the presence of a fish predator, that the N to P of these tadpoles was altered. So there can be these top-down effects on consumer-driven nutrient dynamics. Furthermore, when we're thinking about behavior of organisms, this can alter transformations of nutrients within the sediment layers. So through the movement behaviors of organisms and also their excretion and ingestion, we might have differences in nutrient transformations occurring via these indirect effects. So how do we actually examine whether or not these effects are important at the ecosystem level? So I'm using this framework to think about how we can scale from organismal traits, so things such as body size, their tissue composition, to populations and interactions between populations. So thinking about the age and size structure. Are these organisms moving across different habitat types? To then influence these ecosystem processes. So I'm going to kind of move across this conceptual diagram to think about how organismal traits can be mediating these population and ecosystem level effects. So let's first start with these organismal traits. So we can think about the body size, the trophic guild, tissue stoichiometry, et cetera. So I'm only going to have time to highlight a few of these things. So this paper just came out in Ecology last year, and this is work by Mike Vanny and Pete McIntyre, and this is examining excretion rates as a function of body mass, and also whether or not they're vertebrates or invertebrates, and also looking across temperatures. So these rates vary due to all of these effects. So you see that body mass is a really important attribute, but also whether or not they're vertebrates or invertebrates. And as I mentioned before, these rates scale with temperature. Furthermore, this excretion into P was most strongly driven by the dry mass or the body size of the organisms, and not so much with the body into P. But this is compiling studies across the marine literature and also the freshwater literature. So if we try to examine what's going on within various studies, remember that these excretion rates are also very dependent upon what the food resources are. So if we look within studies, we can see that there are patterns. So Bob showed a figure from this study as well showing that the log in the N to P excretion rate does scale as you would predict by the tissue N to P. And some work I'm doing with freshwater mussels also shows this same pattern. And there's also a lot of variation due to age and phylogenetic grouping that I don't have time to get into today. But these are some considerations that you need to make when you're thinking about how these organismal traits might affect this nutrient recycling within systems. Moving into the marine world, so I do have a few examples of that. So this is some work by Jake Allgaier from an ecology paper in 2013. And so this is herbivores, and these are predatory fish. And so you can see that their N to P varies based on the feeding guild. So you also need to understand something about how these organisms are feeding to make predictions regarding what they're excreting and how they're making nutrients available. So that was really quick, but I only have so much time. So now we got to think about the population effects. So we need to think about what is the population size of the organisms that we're studying, what's their age and size structure, because there can be a great deal of variation in what they're sequestering in their bodies and excreting or adjusting to the system, and trying to understand what's their distribution. Are they clustered, or are they randomly kind of spatially distributed? Are they migrating? So these are really important things to think about when you're trying to understand the importance of these consumers. And the nice thing about this is this provides a framework where we can compare across systems. We can compare across species. So this is just a few studies that have been done in freshwater systems. And so we can actually, we can start to, if we understand the distribution and the biomass of these organisms, we can look at how much nitrogen are they excreting per meter squared per hour. And so we can compare across systems and within systems across organisms. And so going back to some of the work by Jake Allgaier that he's done in these reef systems, he showed that herbivores versus predators, well, the predators, they're contributing a lot more nutrients to the system. And then as you change who's contributing to the biomass across different systems, so we can scale this up and think about how much nitrogen, grams per meter squared, are they storing? And also, what are they excreting as far as grams per meter squared per day? And so we can compare across all these different system types and see who are the big players, what's contributing to the nutrients that are readily available versus what's being sequestered. And what he found in this study is that this is really dependent upon the age or size structure of the population and who is there. So species are important, and also the age and size structure of those populations. And so some work I'm doing now, so I'm also gonna kind of go off on some wild tangents and hoping to get some feedback on some of the stuff I'm doing now. I'm doing some work in which I'm looking at a very biodiverse community and trying to understand how organisms are functioning in these systems. So I'm trying to do some spatially relevant quadrat sampling in which I take these GPS coordinates, digging quadrats in the stream, and measuring excretion and digestion rates, and also tissue stoichiometry across these sites. And in these sites, have a lot of variability, high species richness, very high densities in some cases. And so I can take this data, here's just the individual species excretion rates, but I can take this data and kind of scale it and think about, okay, here's my densities. This is based on a kernel spatial interpolation in ArcGIS, and I can think about the densities within these different sites and also scale my nitrogen and my phosphorus excretion data to this. And you can see, there's a lot of correlation here, but it's not a direct correlation. And so if I dig into these data further, some species are doing more than what their biomass would suggest. So if I look at the proportion contribution to biomass versus the proportion contribution to something, let's say, nitrogen excretion, some species are playing a larger role. And then if we just look at the N to P of excretion of the average is about 18.2. In comparison to the background water column, which was 8.9. So this suggests that these organisms might be alleviating nitrogen limitation in this system. And then kind of continuing further down the rabbit hole, trying to think about this, do species matter? Do we need to, can I just apply one value? Like all of these mussels are in the same family, but do I need to know anything more? So if I just apply an average excretion value for this whole community versus what my real values are, we can, using the raster calculator and ArcMap, we can construct these maps. So yellow is essentially where I'm saying there's no real difference versus the red and orange, I'm overestimating what these rates are, while the blue is an underestimation. So if we're looking at in excretion, I'm doing good with this average value for a mussel, about 43% of the time. But I'm overestimating it about over half the time. If we look at phosphorus, I'm doing a really good job. So 90% of the time, I'm right on. And then if we look at the N to P, we're doing good about 65% of the time, while overestimating this over 30% of the time. So this begs the question, how much do we need to know? Do we need to know about species? So you guys can talk to me about that later. So with the effects on ecosystem processes, so I beg you guys to think about how you can apply this to your system, because I'm very much in my stream or my little wetland, and you guys work in a really big place. But think about how these organisms might be altering things like nutrient limitation, production in your system, decomposition, and energy flow. Energy flow, those higher trophic levels. And I'm going to tell you a little bit about how other people have been doing this across systems. So, you guys are always thinking a lot about the biological pump in oceans, and this work by Roman and McCarthy really talked about the whale pump and moving these nutrients back into the photic zone, and I just really thought this was an incredible piece of work. And within streams, we think about these kind of biogeochemical hotspots these animals might be creating. So these areas that have disproportionately high rates of reactivity relative to the surrounding matrix. And this work by Pete McIntyre and others showed that due to the aggregating behaviors of these fish and their habitat preferences, that you had these areas of biogeochemical hotspots of nitrogen and phosphorus excretion. And they altered the relative availability of N and P. And he showed by measuring nutrient uptake in the system relative to what these organisms were excreting, that these fish could meet up to 75% of the nitrogen demand within the system. Furthermore, there's also a lot of temporal variability. So in this work by Krista Capps and Alex Flecker, they showed that these invasive fish could create these biogeochemical hotspots, but it was temporally variable. These animals create these aggregates during the day, and then they move away from one another at night. And so during the day, they were creating these aggregations and creating this temporally and spatially variable hotspots of biological activity. Some work that I've done in the past, this is some work I did in Oklahoma. I mapped the distribution of unionid freshwater mussels in a river, mapped out 47 mussel aggregates within this system, and measured the excretion rates. And you can see as you move down the system, we have these areas where there's high densities of mussels, and you get these hotspots of nitrogen and phosphorus. And cumulatively, you have a lot of N and P being excreted. I want to focus here where we have the excretion into P. You see variation in the N to P that's available. So that's due to the fact that you have different species that have different requirements for nitrogen and phosphorus. And so this particular species, Actinoneosis, it's different than most of the other organisms, and it's also classified as thermally sensitive, which I'll get back to at the end of my talk. And these areas that had the higher N to P were dominated by this particular species. So as you change species composition, that might alter your nutrient availability within a system. Also, we need to think about sequestration. So organisms are growing, they're taking up nutrients, and so in my system, these organisms live a relatively long time, so anywhere from 5 to more than 50 years. I've aged these things, have 90-year-old individuals, and they're sequestering a lot of nutrients in their shell and their tissue. And so by looking at their growth rates and also what their requirements are, we can think about how much nutrients these organisms are sequestering. And when we're scaling this up, we can also measure things like ecosystem metabolism. And so in some work that I have coming out, I showed that these mussel aggregations are driving areas in which they have higher primary production than areas without mussels. And is this actually important? So I've talked a lot about excretion, digestion, how much nutrients these things are storing, but so? Well, what does it matter? Well, some of the work I've done has shown that these organisms can actually alleviate nutrient limitation. So in sites with areas without mussels, We had nitrogen limitation as measured by nutrient diffusing substrates. And the systems were co-limited by both N-to-P and areas with mussels. And we also saw a difference in benthic algae species composition. So sites with mussels, which are these black dots, were more dominated by diatoms, while sites without mussels were more dominated by blue-green or cyanobacteria. So this suggests that there's shifts in community composition due to this alleviation of nitrogen limitation. I'm sure you guys all know that these blue-green algae are N-fixers, so in these sites that are N-limited, you had a higher dominance of these blue-green algae. We can also trace this kind of stuff into the food web. So I used N15 to label mussels and track this nitrogen into the food web. And so I enriched mussels. I grew them for about over a year and placed them out in a stream system and sampled the food web biweekly for an entire summer. And as well, I conducted nutrient uptake to look at the nutrient demand of the system in comparison to what these organisms are excreting. And just cutting to the chase, these organisms could be, based on my results, could meet up to 40% of the nitrogen demand. And in a natural mussel community where we have really high densities, could have been up to 98%. And with the N15, with the tracer results, up to 70% of the nitrogen in the food web, in those organisms that I sampled, were from mussels. So you can trace these things and try to think about how important this nutrient recycling is. So kind of going back, biodiversity and ecosystem function and thinking about how changing environments might alter this. What kind of results do we have so far? Think about climate change, fishing pressures, et cetera. So kind of going back to some of the work, Jake Allgar's worked a lot with Craig Lehman. And Craig, he did some work where he compared sites that have been fished versus unfished sites and found that the nutrients that are available by fish, of course, are, there's more nutrients being recycled by fish in these unfished sites. But also, as you get fragmentation of these mangrove systems, you're also seeing an impact where there's less fish, and so less nutrients being recycled by these organisms. Furthermore, this work by Dan Spooner has shown that, as I showed before, these consumers are leading to greater nutrient heterogeneity based on their spatial and temporal aggregations. And this work by Dan Spooner shows that as we have greater nutrient enrichment to the system, this is kind of dampening out that effect where these organisms aren't going to be influencing and enhancing heterogeneity of our ecosystems. And as we lose species due to more drought events or and things of that nature, we're seeing loss in species that are perhaps thermally sensitive. So in this work that Heather Galbraith did and some work that I followed up on, we're seeing declines in the relative abundance of thermally sensitive species. And if you recall, one of those thermally sensitive species had an interesting stoichiometry in which they're excreting at higher N-to-P. And due to this drought, we saw huge declines in the amount of nutrients that were being excreted or remineralized and losses in the storage of these nutrients. And due to this drought, we saw kind of these shifts in excretion and what the aerial tissue N-to-P was. And so this kind of gives us a framework to think, okay, can we make any kind of predictions? Like, is it all lost, or can we gather any sort of information from this? And so this is going to be my wild tangent for this talk, and asking, can we think of stoichiometry and stoichiometric traits And are they varying as a function of any sort of sensitivity that we might have to systems? So whether it's thermal sensitivity or sensitivity to any sort of nutrient pollution and things of that nature. So in my mussel system, vulnerability to this extreme drought event was linked to some of the body stoichiometry, and that reduced nutrient recycling, and we also saw a change in the N to P ratio that was being excreted. So can we use this kind of framework, these stoichiometric traits and these physiological traits to understand ecosystem vulnerability in this kind of state of change that we're seeing, to predict species losses and consequential declines in ecosystem function? So we can think about this vulnerability to climate change, and is that directly related to any sort of to body stoichiometry or not? I don't know. And how does that translate to ecosystem level effects? So we can make some kind of hypotheses and predictions regarding this. So if we're thinking about the vulnerabilities to some sort of environmental perturbation and a specific trait, if there was going to be no effect and species are just lost, maybe we're going to be okay if we just lose these species because we might still maintain that function within the system, right? So it'd be just along this red line. But if vulnerability and a specific trait such as body or excretion stoichiometry, if they're related to one another and we lose these organisms up here, we're going to lose an entire piece of that system. And so if we can understand if these traits and functions that may be lost, whether or not they're correlated to other, we might be better able to predict the ramifications of ecological change in our systems. So that's going to be my wild tangent for this talk. So can we kind of put this together? And I'm going to continue down this rabbit hole of thinking of consumer-driven nutrient dynamics to try to answer these questions, because I think it provides a nice framework for thinking about from the organismal level all the way up to that ecosystem level scale to think about how biodiversity change might alter ecosystem functions in our changing world. So kind of putting this together, the physiochemical structure influencing community structure, which might alter kind of the species traits that we see in that environment. And so we can use ecological stoichiometry as a framework to think about what species we have in our systems and the traits that they possess and how that influences various ecosystem processes. So, I think it's quite an elegant framework myself, and ecological stoichiometry was like my bedside reading when I was in grad school. So anyhow, I appreciate your attention, and if you have any questions, I will do my best to answer them.
Do you have questions?
So in the Great Lakes, when the zebra mussels came in, they changed some of the, they cleared the water, but they changed some of the algal communities because of their excretion of, I think it was ammonia. So you tend to talk in CNPs, but and Pat will probably talk on this. There's different kinds of N. So can you address maybe what the mussels excreted and how that affected the algal community?
Yeah, of course. So in the system I work in, or the system I spend my most time in, is with native freshwater mussels, and with my work with them, it's primarily ammonia. So that's the vast majority of what's being excreted, and they have a lot different characteristics than zebra mussels in which they bury into the sediment. And so oftentimes you'll just see their siphons right at the water sediment interface. And so I think they're having the largest effect, kind of on those benthic algae, and some of it's getting redistributed in the water column as well. But I think, from a stream ecologist perspective, you have that unilateral direction of flow, and so those nutrients get suspended in that water column and get moved downstream, and we often think of nutrient spirals and how nutrients are getting taken up with that unidirectional flow of water, and so it's just getting taken up by the benthos very quickly in these kind of relatively shallow water environments, especially when you guys are talking about like kilometers and depth.
Yeah, thanks, Carla. It's really great talk. The relationship between vulnerability and N to P ratio, kind of near the end there, was fascinating. And I wonder if you could elaborate a little bit more on what that connection is. At some level, just observing that thermally sensitive species tend to have a certain N to P ratio. Okay, that's a sort of a statistical fact, but have you been able to come up with ideas about what's driving that, what the mechanistic connection is? Why would a thermally sensitive species have a different N to P ratio than a thermally robust species does?
Yeah, I've spent a lot of time trying to think about that sort of thing, and with the experiments that we've been doing, measuring Q10 rates and things of that nature, it just seems like these thermally sensitive species, once you reach a threshold, they start catabolizing their tissues, and these tend to be the species that have a lower N to P ratio, and I think they're kind of running out of that N reserve more quickly or something. At least that's what I'm seeing, because it tends to be the more thermally sensitive species that have a lower N to P and are excreting at a higher N to P, and I think they're kind of getting to a point where they're almost unlimited due to the fact of the catabolizing of tissue.
Thank you very much.